And for our first speaker, I'm very excited to introduce Ximena Garcia Arceo. She's a chemistry graduate student in the ZID Lab, here to present her work. And with that, I'm happy to turn it over to Ximena, and she can share her screen.
Thanks, Evan. And thank you to all the DAZZLE organizers for inviting me here today to talk about mRNA-mitochondrial localization in simulated cerevisiae. This is all about my computational research into mitochondrial regulation in Saccharomyces cerevisiae. So that was a little bit of a pun there. So firstly, I want to talk about how I grew up in the San Joaquin Valley. This picture here was taken behind my parents' home. We're surrounded by fields, and I grew up that way in a very rural area. It's right in the middle of California, and it's also, the area also has a lot of immigrants and first-generation Americans, particularly with Latinx backgrounds. And so a lot of people are immigrants, a lot of people work in the fields, and that describes my family very well. So I always felt like I was representative of the area that I grew up in. And that sort of led to a culture shock when I went off to college at UC Santa Barbara, where I then majored in physics. So that just amplified the culture shock even further. So first I want to show a plot of some figures that I had no idea about. Firstly, the representation of women is lacking in physics. That's no surprise to anyone, but to look at the numbers, and if you look at the most recent numbers in 2016, it's about 1 in 6 women, about 1 in 6 bachelor's degrees go to women in physics, and it's a little bit better than what I saw at UC Santa Barbara. So again, this is a national statistic. Another national statistic has to do with the representation by race and ethnicity. So people that are labeled Hispanic American make up about 7% of all the physics bachelors being granted in the United States. UC Santa Barbara is actually leading in this area. According to the AIP, it's one of the schools that grants the most physics degrees to Hispanic Americans, which is about 10% of graduating class is Hispanic American, and that's considered leading and being one of the better schools at representation and retention in the field. So I realized that was kind of as good as it was going to get by being at UC Santa Barbara when it came to inclusivity. And actually spending the last 2 years there at the Materials Research Lab was not just formative educationally, but crucial psychologically for me because they were so inclusive in the Materials Research Lab. And I can't thank them enough for transforming that campus for me and making me feel welcome in my field and competent in my field, something I hadn't felt in my first 3 years. Actually, and then I came to UC San Diego still as an undergrad, thanks to UCSD STARS. UCSD STARS is one of those programs that helped me forge academic and professional networks. And at UC San Diego, I also found a very supportive environment there in Professor Koslover's lab, still in the physics department. I started prioritizing I started prioritizing the environment that I work in a little bit more after I realized how crucial the MRL at UC Santa Barbara was for me in terms of feeling welcome and feeling like I can actually do work and like I'm capable of doing work. So here's a list of research programs that are either UC-wide or national that I benefited from and that I would encourage people to look into if they want to help other women and ethnic minorities in the sciences. And the people that I worked with at UC San Diego as an undergrad, we're actually still working together. There's a collaboration between the ZID Lab and the Koslover group still going on. And it's a collaboration of physicists and chemists and biologists. And one of the open questions in biology we wanted to explore has to do with the Warburg and Crabtree effects. So in this picture here, it describes the Warburg effect. So that's when that's the observation that tumor cells are turning their sugar into lactate through aerobic glycolysis. So even though there's plenty of oxygen around, these tumor cells prefer to use glycolysis, which is very inefficient. And even healthy yeast cells will do the same thing, partly because aerobic glycolysis is just faster. And what we wanted to do was build a quantitative predictive model of metabolic regulation in eukaryotes by starting by looking at the mitochondrial regulation and function in Saccharomyces cerevisiae. So using yeast to tell us a little bit more about these higher eukaryotes. So yeast will switch their metabolism from fermentative to respiratory when sugar starts running low. And now respiratory flux starts picking up when sugar is low. And mitochondria size also increases. So there is simultaneously, or nearly simultaneously, a change in metabolism and a change in mitochondrial morphology. So mitochondria are needed for cellular respiration. So that just sort of clarifies the connection there between metabolism and mitochondria. But what's unclear is whether there's a post-transcriptional mechanism for this metabolic switching, or at least what that mechanism may be. Some transcription factors have already been implicated in metabolic regulation and metabolic switching. But what we wanted to do with our model was incorporate all the data about what's going on post-transcriptionally with genes and the changes in mitochondria, so all under one model that could be predictive and applied to other eukaryotes. One way we think that the mitochondria is responding to cell needs and reading the cell state is through mRNA localization, and other labs have also suggested that this could be one method for mitochondria to communicate with the cell at large. And localization is important for co-translational import. So it's especially important for membrane proteins like mitochondrial proteins. And every gene tends to rely on just one primary import mechanism. So there's additionally a question of how localization is attuning, or rather how localization can be accomplished. So localization is important for the co-translational import, just shown down here in pink. And that's the process by which an mRNA is being translated simultaneously with being localized to the mitochondrial surface. And co-translational import has been observed for a lot of genes that are very rich in transmembrane domains. So there's a hypothesis that co-translational import and localization are just important for protein folding, but additionally, we think that this could have a role in metabolic function because the proper import and function of mitochondrial proteins would be very important for getting mitochondria up to a bigger size so that it can respond to the cell needs. So there's an interplay there between what's going on molecularly and system-wide. And these localization trends, again, they are gene specific. mRNAs that have been labeled as constitutively diffuse usually code for outer membrane proteins. So these mRNAs are for like TOM22 and also the gamma subunit of ATP synthase, which is very important for the respiratory chain. So it's important to understand how those mRNAs and those proteins get to their final destination. And then there are constitutively localized mRNAs, which includes TIM50, and those tend to code for intermembrane proteins. And again, there is a trend there that has to do with just the protein. The protein itself, since they tend to have a lot of hydrophobic residues. So we wanted to understand the mechanism of localization. And something that starts to color in that picture for us has to do with an experiment done by the Weissman lab in 2014, where they found that completely inhibiting elongation promoted localization for about a quarter of the genes that code for mitochondrial proteins. So genes like ATP3 are considered constitutively diffuse until their elongation is inhibited. And now those mRNAs suddenly start preferring to localize to mitochondria. So we're very interested in what's going on there and why a quarter of the genes seem to have this switch-like behavior in their localization, whereas about another quarter of the genes are going to localize without any sort of sensitivity to elongation, and about half of them cannot be made to localize, cannot be targeted to mitochondria by arresting their elongation. So this interest, having three classes that all respond differently to elongation inhibition, gives us a clue to how localization is accomplished in the first place. And what that Weissman lab, what they suggested in that paper was that completely inhibiting the elongation is increasing the nascent peptide exposure, and that is what is helping what's helping with co-translational import. So some genes don't have any known mRNA binding partners, and we think that these genes are must be relying on the nascent peptide to mediate the interactions between the mRNA and ribosome complex with the mitochondrial surface. So by having that nascent peptide exposed for a longer period of time, the mRNA and ribosome complex have themselves more time to localize to mitochondria. And the fact that there was those genetic trends that I showed in the previous slide suggests that a mitochondria targeting sequence is necessary, but it's not sufficient for localization, and that's why genes with a switch-like localization, which includes ATP3, can only localize if elongation is inhibited. If elongation is not inhibited, their MTS will naturally appear and disappear too quickly for the nascent peptide to facilitate localization. So Tatsu is a postdoc in my lab who wanted to look at localization in a more quantitative way. So he defines the ratio of localization as the time an mRNA spends near the mitochondrial surface compared to the total mRNA lifetime. And what he found was that the so-called constitutively localized mRNA, TIM50, shown in blue, it does have a pretty high ratio of localization. That's that blue bar, and TIM50 spends over half of its time near the mitochondria. What's interesting is that TIM50 contains ribosome stalls, which suggests to us that TIM50 elongation is slow. So again, it's consistent with the hypothesis on elongation in the literature. And his ratios of localization for constitutively diffuse mRNAs, ATP3 and TOM22 were also consistent with the literature. We see that their localization is pretty low compared to this blue bar, but there's even some variation there between these two mRNAs with the same label. So that was interesting to see, and again, it's all about getting a quantitative picture so we can simulate it. And what he found was that the ratio of localization varied not just by the gene, but by the metabolic state. So by switching the metabolism of the metabolism of the cell from vegetative or fermentative conditions to respiratory conditions, localization increased for all the mRNAs, even this orange one on the end that's called constitutively diffuse, TOM22. So now that we have all this information on how localization varies by gene and metabolism, that's what we're going to put into our model. And what was interesting was that the mitochondria size also increased. So maybe the ratio of localization depends not so much or not only on metabolic state, but also on mitochondrial morphology. So again, this is bringing more quantitative data into the picture, which makes my life easier so that I can simulate all this stuff. And what Tatsu did was look at mitochondrial volume fraction and how that's changing. And he defines that as the ratio of the mitochondrial volume to the free cytoplasmic volume. And what he found was that increased mitochondrial volume fraction is associated with increased localization for all three of the genes that we're looking at, which again correspond to those three classes of elongation dependence. And the curves trend upward for all three of them, but it's the shape of them that's really interesting. In this paper that's going to be published soon, we looked at the qualitative behavior of TOM22. We found that corresponds to the behavior of a randomly diffusing particle. So as mitochondria get bigger, diffusion alone can bring more mRNAs to the mitochondrial surface. The mitochondrial surface just becomes easier to find if there's more of it. And this is seems to explain TOM22's increase almost completely, but it doesn't explain why ATP3's ratio of localization increases so much with respect to mitochondrial volume fraction, and it doesn't explain why TIM50's ratio of localization is high throughout. And we think that the MTS interactions with the mitochondria can maybe explain the rest of this difference that we see in the green and blue curve. And this is something that wasn't included in this paper. So this is all the MTS interaction data and simulations are new things that we explored in a kinetic model. We just had, in our simulation, we had the rule that only mRNAs with an exposed mitochondria targeting sequence can bind to the mitochondrial surface. So we just simulated translation kinetics and diffusion. So I'll talk about the translation kinetics first. Translation is just 3 steps, or we simulate it as just 3 steps. Initiation, which occurs at a certain rate, and elongation and termination, which occur at their own rates. And these numbers can either be calculated and approximated, or in the case of initiation, found in bioinformatics papers. So that's how we were able to get that for our simulation. But this differs a little bit from the typical picture of translation in that we have a mitochondrial targeting sequence shown in pink. And so a ribosome initiates on an mRNA, but that alone is not enough to give the mRNA an affinity for the mitochondrial surface. The ribosome needs to chug along far enough and elongate far enough to expose the mitochondrial targeting sequence. The ribosomal tunnel is of a finite size as well, so that already introduces a physical constraint to how far along a ribosome needs to be on an mRNA for that mRNA to preferentially localize to mitochondria. Without this sticky pink nascent peptide, mRNA will just diffuse away. So here are my 5 simulation inputs. The mRNA length and initiation are things that can be found online. Elongation and termination are usually equal to each other. So if you know the elongation rate, you know the termination rate. And the 5th and final simulation input I'll talk about has to do with the mitochondria volume fraction. So we described and simulated the mitochondrial morphology by creating this very sort of abstract cell. This cell is actually a set of cylinders, of tubes. The innermost cylinder has the same physical size as the mitochondrial radius, and the outermost cylinder describes the rest of the cell. So it's the ratio of the cytoplasmic radius in the simulation with the mitochondrial radius that describes the mitochondrial volume fraction. And then we let the mRNAs just diffuse around this cell. They can't leave the cell, they can't go into the mitochondria, so they're just bumping around between these two cylinders. And a randomly diffusing mRNA without an MTS, or that's very far away, is shown in white. So as you can see, you can have mRNAs close to the mitochondrial surface just through diffusion alone, and that's what we see with TOM22. TOM22 only interacts with, it doesn't interact with mitochondria. It's only near mitochondria because of diffusion, whereas the other 2 genes, like ATP3 and TOM22, will stay on the mitochondria longer because they sometimes have an exposed MTS. So they'll stick around the mitochondrial radius and contribute a lot to localization. So when I initialize my simulation, the mRNA is born very far from the mitochondrial radius, and it's diffusing simultaneously with ribosomes elongating and initiating, and it just diffuses around until it finds, until it gets close to the mitochondrial radius, and then the simulation checks whether there's an MTS exposed. And if it is, which is shown in pink, the mRNA will stay, but it'll only stay for as long as the MTS is exposed. Once it's white again, it leaves. So that diffusion brought the mRNA towards the mitochondria, and once there's no molecular reason to keep the mRNA there. It takes it away from the mitochondria, because that's the thing about diffusion. Diffusion, it's completely non-specific. It'll move things every which way. And I think it may be my physics background. I'm always interested in how much a cell can accomplish through just diffusion alone, because diffusion is free. So what we saw was when we looked at localization, was that longer genes will probably have a higher localization. This is also consistent with experimental data, but I just want to go through what the inputs tell us. And elongation, it does set the localization ratio value, but more importantly, it seems to set the shape of the curve. And it's the shape of the curve that tells us about mechanisms. So it was important to explore what's going on with elongation. And then initiation rate doesn't really set the shape of the curve. It just sets the localization ratio value. So taking all three of these inputs that define a gene into consideration, I was able to recapitulate the curves from experiment with just data that we could find online or calculate back of the envelope. So it's a very simple model with a few assumptions and not a lot of molecules, but that I was able to recapitulate a localization curve. And going forward, what we want to do is apply this to other genes and then go into the lab and experimentally test their localization and their elongation rates. So in conclusion, we find that to increase the localization ratio, we have a gene would probably need to be long. It would need to have a slow initiation and a slow elongation. To increase the MTS exposure time, which is important for a reason I'll get to in a minute. Long genes and slow elongation and slow elongating genes have a higher MTS exposure time, but a high initiation also leads to a higher MTS exposure time. So there's a little bit of a competition between increasing your localization and increasing your MTS exposure time. So it's not a one-to-one relationship between these two things. And indeed, as you can see here, on the right hand side, there's the normalized MTS exposure time in orange and the localization ratio in blue, and they don't exactly track each other throughout the range of mitochondrial volume fractions. But MTS exposure can explain or can predict localization ratios when mitochondrial volume fractions are big, but exactly where that switch in control between diffusion and MTS exposure happens is still, it depends on the combination of genetic parameters like length and initiation. So diffusion is really important when mitochondria are small, because it takes so long for the mRNA to reach mitochondria. Whereas when mitochondria are really big, the mRNAs take less time to find the mitochondria. They have less trouble finding mitochondria. And now they're limited by how sticky they are and how often they're sticky. So translation kinetics are important, are especially important when the cell needs to switch to respiratory conditions. And translation kinetics are something that the cell can control or regulate by changing initiation and elongation rates, which we know are which we know can be controlled by yeast. And we want to know moving forward whether initiation and elongation rates can explain what's going on with genes that are conserved in eukaryotes, including the genes in ATP synthase, which are so important for proper respiration. And it seems like almost all of them may be under this control. So cyclohexamide inhibits elongation. And we see that in this very last column that says yes, almost all the way down, inhibiting elongation promoted or induced higher localization to mitochondria for these genes. So this is hopefully going to start to help us complete the picture of how the cell is using mRNA localization to control or regulate its metabolism. And I want to thank my lab. It's Brian Zib lab, and Tatsu is the one that did all of those very great quantitative detailed measurements on localization in mitochondrial volume fraction. Juan Fu has started or has been getting some really great information on elongation of these genes. And then our collaborators are in the UCSD physics lab, and they were instrumental for getting this model and the math up and running. Okay, thank you.